Okay, so good morning, everyone. Today we will have our discussion for General Biology 2 for week number 1. But before that, let's start with a Bible verse for the day. So, do not conform to the pattern of this world, but be transformed by the renewing of mind. Then you will be able to test and approve what God's will is. His good, pleasing, and perfect will. For Romans 12, verse 2, for reflection, the verse reminds us that when we feel afraid, that fear isn't from God. Instead, God gives us three powerful gifts: power, love, and sound mind. The power that God gives isn't about giving us strong on our own, but about having the strength to face challenges with His help. When we're tempted to let fear control us, we can remember these gifts from God. They help us overcome our worries and live boldly for Him. This verse encourages us to trust God's strength rather than giving in to our fears. For our prayer, Heavenly Father, thank you for not giving me a spirit of fear when I feel afraid. Help me remember the power, love, and sound mind you've given me. Fill me with your encourage to face today's challenges. Help me think clearly and act lovingly, trusting in your strength rather than my own. In Jesus' name, amen. Okay, so let's start our discussion. For week number 1, we're going to discuss the reproduction, development, and nutrition. Here are the objective of our module number 1. So at the end of the lesson, you are expected to differentiate asexual and sexual reproduction. Second, describe the different mode of sexual and asexual reproduction in plants and animal. Define the nutrient and cite the nutritional requirements for plants and animal. Trace the pathway of food processing in mammalian or specifically human digestive system. And lastly, for your performance test, you will create a poster or drawing that represents the interconnectedness of autotrophs, heterotrophs in terms of their nutrition. And lastly, you're going to demonstrate an appropriate appreciation for the interdependence of plants and animals in the ecosystem. So that will be our objective. Here are the essential questions that you must answer after our discussion. So, from the smallest single cell organism to the largest whale, all living things shares the fundamental processes of reproduction and development. Why these processes are essential for the survival and continuation of life on Earth. The second question: how do the nutrients you absorb from the meal contribute to your overall health and well being? So, your answer will be posted on the comment section, and then I will check your answer. Here are the terminologies that you should be familiarized first before we proceed to our discussion. The first one is offspring. Offspring, the product of the reproductive process of plants or animal. Second, nutrients, any substances required for the growth and maintenance of an organism. Third, autotrophs. Autotrophs are organisms that obtain energy from sunlight and chemicals to produce their own food. Heterotrophs, those are the organisms that cannot make their own food and obtain energy from other organisms. Next is asexual reproduction. That is the mode of reproduction that does not involve the use of gametes or sex cells such as sperm and egg cells. Next is the sexual reproduction. That is the mode of reproduction that involves the use of gametes or sex cells. And then last one, gametes are the organism's reproductive cells or sex cells, including egg and sperm. So, egg for female and sperm for male. So, let us define what is a reproduction. So, reproduction is a process of the continuation of life here on Earth. So, we have two types of reproduction, the asexual. So, asexual reproduction result in any new organism formed by mitotic cell division, producing an offspring that are genetically similar to their parents. So, here in the process of asexual reproduction, they doesn't need or it is not required for them to have a partner, uh, and most of their genetic material are identical to their parent cells, unlike the sexual reproduction. So, sexual reproduction, it is a process wherein it involves the fusion of gametes or sex cells from two parents or parent organism which are reproduced through meiosis. So here in asexual reproduction, we uses this mitotic cell division, and then sexual reproduction, we uses meiosis cell division. So let's start with asexual reproduction. Okay, asexual reproduction in single cell organism and other animals, mitosis is also their form of reproduction. So it is not just limited by uh, binary fission. No? They also uses mitosis. This does not require fusion or gametes or other sex cells in order for them to reproduce an offspring. Instead, their parent cells form a group or clone for this process, and each multicellular organism can also capable of reproducing through asexual reproduction. So as you can see here in our PowerPoint, it shows a simple representation of how asexual reproduction happens under the mitosis cell division. Under asexual reproduction, we have different category. First one is fission, followed by fragmentation, budding, and lastly is for the vegetative reproduction in plant. The first type of asexual reproduction is binary fission. Binary fission is a type of asexual reproduction wherein the parent organism, such as a unicellular organism, splits into two daughter organisms. So this type of asexual reproduction is usually done in prokaryotic cells such as bacteria. So as you can see here in our PowerPoint presentation, that is an example of amoeba that undergo a binary fission to produce its identical daughter cell. So um, the binary fission is one of the simplest way on how bacteria reproduce easily. No, from one parent, they will elongate their nucleus by copying or duplicating their genetic material, and then the division of nucleus as well as the cytoplasm started to create two daughter or two identical daughter cells. The next asexual reproduction is fragmentation. Fragmentation is considered to be asexual reproduction when it occurs it occurs in a parent organism that break into fragments or pieces. So each fragment from the parent will develop into a new organism. One good example for fragmentation is starfish. Okay, so as you can see, this leg or arm of a starfish are recently decapitated, and then after on they didn't die. No, they create a new organism. So that is a fragmentation. The third one is budding. Budding is a type of asexual reproduction wherein the organism is reproduced by forming an outgrowth or known as a bud from a parent's body. And one example for budding asexual reproduction is hydra. So the parent hydra will form a bud, and then this bud will develop into a younger hydra until it reaches the maturity to detach itself, and then that one or that new organism is the daughter of the hydra. So the sexual reproduction budding. Okay, so this is the process of budding. Next is parthenogenesis. Parthenogenesis is the development of an embryo from unfertilized egg. So one example of organism that uses parthenogenesis parthenogenesis are some insects, reptiles, and fishes. So it is a form of asexual reproduction where female can reproduce even without the male to mate with. And then let's proceed to the asexual reproduction under plants. So that is known as vegetative reproduction. So it occurs when new individuals are formed without the production of seeds or spores. We all know that seeds can be formed when the pollen of a plant fertilizes the ovary. So we have the male gamete and female gametes for plants and animal. So if we doesn't use the two gametic cells, we are using the vegetative reproduction. Here are the examples of asexual reproduction under vegetative propagation. The first one is runners or stolon. Runners or stolons are described to have a longer, slender stem that grow along the surface of soil. So here, runners and stolon doesn't use seed for their reproduction, but they use reproductive stem. So their stem can be found on the surface of the soil, and then once it was matured, it will create a node to create new plant. One example, some example of runners or stolons are gabi, strawberries, and water lilies. Next is suckers. This vegetative propagation is roots that could give rise to new plants. They uses reproductive roots. So here are the examples of plants that uses roots for them to create new plant or bamboos, bananas, and cherries. Next is rhizomes. Rhizomes are underground stems that grow horizontally so they can reproduce new plants at nodes along the rhizome. So if the stolons or runners, their reproductive stem can be found on the top of the soil. Rhizomes, we can find their stem under or beneath the soil. Grasses, ginger, and sedges are example of rhizomes. So as you can see, once their horizontal stem uh, grow under the soil, it creates a nodes to form a new plant. So it is a type of asexual reproduction under plants. Next is corms, bulbs, and tubers. So here, this plant uses a modified stem that store food reserves so they can reproduce new plant from their buds within the structures. So as you can see, potatoes, yam, onion, and tulips are example of tubers. So how can they reproduce? They uses their roots. Okay, so the roots are being swollen, and then those swollen roots become their food storage. Next is sporulation. Sporulation is a uh, vegetative propagation that involves the production of spores, which are specialized in asexual reproduction. One good example of sporulation is ferns. Okay? And then the last one is tissue culture. So most of the examples such as runners, corms, bulbs, rhizomes, and suckers, those are or they can be observed in a farm, a garden, but here tissue culture is an example of asexual reproduction done within the lab. So tissue culture is also known as protoplast fusion. So here, the tissue culture involves growing plant cells in laboratory setting. So the protoplast fusion specifically refer to the merging of two plant cells, which is the protoplast, without cell wall to create a hybrid plant. So here in tissue culture, we will just use no the process of uh, culturing for them to create new plants. So the process of plant tissue culture, the first is the tissue samples will scrape from the parent cell or the any part of their stem, leaves, or roots. And then the tissue samples placed in the agar growth medium containing the nutrients and auxin for the development of their uh, plant. So auxin is one of the hormones releases by plants for growth and development. And then third, samples develop into a tiny plant leaflets. And then lastly, plant leaflets will planted in a compost soil. Okay, so that is a tissue culture. Now let's proceed to the sexual reproduction in humans or in animals. So here, we uses the meiosis, which consists of uh, two gametic cells, the sperm and the egg cells, wherein they consist of two rounds of cell division, the meiosis 1 and meiosis 2, if you remember from our previous discussion. Here, the daughter cells of meiosis are called germ cells, which develop into gametes or sex cells. The egg will came from female, which is usually large and non-motile. The sperm came from male, which is generally smaller and motile. Here is the image that shows the comparison between the two gametes in animal. So sexual reproduction gives the offspring half of the maternal traits and half of the paternal traits, thus creating a unique organism. So the 23 chromosomes will be donated by the mother, and then another 23 will be donated for the from the father. Okay. So, process of sexual reproduction in animals, we have two classification, the external fertilization and internal fertilization. So, let's discuss what is external fertilization. First, uh, fertilization is a process of fertilizing the egg. No, uh, fertilization is a process wherein the sperm fertilizes the egg. We have two classification of fertilization. The first one is external. That is a process when the egg and sperm meet outside the body of a female. So as you can see here in our PowerPoint, uh, this is a common practice among fishes where the female releases thousands of eggs in the water simultaneously, which the release of sperm cells by the male, followed by the fertilization. So the external fertilization is described to be risky and have a lower chances of survival, or lower chances for them to proceed in fertilization. And we have the internal fertilization. It is a type of reproduction wherein the egg is fertilized inside the body of a female. So, for vertebrates that practices internal fertilization, the embryonic development could be through oviparity, uh, ovoviviparity, and viviparity. So, what is the difference within the three internal fertilization? So first is the oviparity. The egg are fertilized internally and are deposited outside the mother's body to complete the development. So one example of oviparity among animals are birds and reptiles. So uh, they produces egg through reproduction. Next one is ovoviviparity. Here, the eggs are retained inside the mother to complete its development, but the embryo gets its nutrition from the yolk. So, some species are snakes and sharks. Next is the viviparity. The young develop inside the body of their mother through the umbilical cord and not from a yolk. So the difference is both oviparity and ovoviviparity uh, uses a yolk. No? They produces egg, while viviparity, this is a process of internal fertilization wherein mother produces a live young one. And most of the animals that uses viviparity for their internal fertilization are mammals. Here are the organs, uh, reproductive organs in human. So, sexual reproduction and meiosis. The sexual reproduction gives the offspring half the maternal traits and half of the parental traits, thus create a unique organism. So, the 50% came from the male reproductive organ. The process known for the production of sperm is spermatogenesis. Here are the reproductive organ of a male. So we have the epididymis, testes, scrotum, the seminal vesicle, vas deferens, urethra, and penis. So all of those reproductive organ is responsible for producing sperms. So these are the sperms. Next is the female reproductive organ. Female reproductive organ is responsible for developing an egg cells, or the process is the oogenesis. The parts of female reproductive organ responsible for developing or producing an egg cell are ovary, we have the fallopian tube, uterus, we have the cervix, cervical canal, we have the vagina. All of those are the female reproductive organ. Here are the stages of mammalian animal development. Once the sperm fused with the egg cell, it is known as the fertilization. So when the sperm fertilizes the egg, they form a zygote. And then after that, it will proceed to the next stage, which is cleavage. Cleavage is a process wherein the zygotes undergo a rapid cell division, forming a balls of cells called blastula. So from one egg, it will become two, will become four, and then so on. So that is the process known as cleavage. They form a blastula. Third one is the gastrulation. Gastrulation is a process wherein the blastula undergoes a series of cell movement forming a three germ layers known as ectoderm, mesoderm, and endoderm. So these layers will give rise to all of the tissue and organ for the developing fetus or organism. The last one is organogenesis. Organogenesis, it is a process where the cells from the three primary layers interact to form specific tissue and organ. So these are the four stages of mammalian animal development. So we have the fertilization that form zygote, and then cleavage that form a blastula. So from one cell, it become two, four, and eight until it reaches the blastula. So that is the blastula, and then it will continue to divide cells to form into gastrula. So gastrula form a three different layer. So here in organogenesis, it is a crucial phase for embryonic development wherein the three primary germ layer known as the ectoderm, mesoderm, and endoderm differentiate into various tissue and organs of the body. So this process begins after fertilization and blastulation. So it is essential for the formulation of a fully functional organism. So the stages of the development of human will start from the formation of a zygote. Okay, uh, a single a single fertilized cell that undergoes multiple of cell division. So this lead to the formation of the blastocyst. Again, so that is the blastocyst. Blastocyst is described to be a hollow spear of cells that implants into the uterine wall. The following is the gastrula. Okay, so that is the gastrula. The gastrula stage emerge marking of the establishment of the three distinct layers. So what is an ectoderm? So ectoderm is responsible for the development of the external body structure and nervous system. So the ectoderm, okay, the purple, yeah, the purple one is responsible for the external body structure and nervous system. It gives rise to the skin cells, the epidermis, pigment cell responsible for coloration, and many more. Additionally, ectodermal cells contribute to the development of germ cell, which is eventually becomes sperm and egg cell. So here, ectoderm are the part of our gastrula that later on, no, it will merge to create a germ cells. which is the sperm and the egg cell. The next one is mesoderm. Mesoderm is also known as the middle layer. Middle layer or mesoderm forms the musculoskeletal, circulatory, and excretory system of our body. So that green layer, yan, that will be later on responsible for the formation of our musculoskeletal, circulatory, and excretory body system. So it differentiates into cardiac muscles, which enables the heart to function. Smooth muscle found in the internal organs, okay, such as the stomach, you know, the skeletal muscle, okay, for our locomotion, for the that facilitates our movement, and mesoderm gives also rise to the kidney tubule cells, which are essential for the filtration, and red blood cells, which transport oxygen throughout our body. Okay, so those are the fate of mesoderm or middle layer of our gastrula. And then we have the endoderm. Endoderm, that is the yellow part of the gastrula. Endoderm, or also known as the internal layer. So the endoderm primarily develop into internal organ lining and associated structures. So it give rise to the lung cells, which is the alveolar cells, play a vital role in respiration. Also give rise to the thyroid cells that regulates our metabolism, and pancreatic cells. Pancreatic cells are essential for the digestion and insulin production in our body. So these cells ensure the proper functioning of vital physiological process within our body. So the significance of organogenesis is highly regulated process crucial for the formation of a healthy and functional organism. Okay, so all living things need energy to survive. So most of our organism directly or indirectly depend on the sun radiant energy for food. Uh, once an organism once an organism need nutrition, they have two fate, being an autotrophs or heterotrophs. Autotrophs are organism that produces their own food. Simply, there are mostly plants, algae, phytoplanktons, and some photosynthetic bacteria. On the other hand, just the heterotrophs, they are consumers. So, the heterotrophs is an organism that does not make its own food, such as animal, fungi, protozoa, and bacteria. We have a mixotrophs. So mixotroph, it is a type of organism wherein the nutrition could be in two modes, such being a both autotrophs and heterotrophs. So a plankton that can use phototropy and phagotropy to acquire nutrition, growth, and reproduction. So as you can see, this organism, they have the chlorophyll that captures the sunlight to create their own food. At the same time, they use phagocytosis to acquire nutrition. So here are the examples of mixotrophs. Let's discuss what is an autotrophic nutrition. So the word autotrope is derived from two words. Auto means self and tropy means nutrition. Hence, autotropic means self nutrition. So in autotropic nutrition, the organism produces its food from inorganic raw material such as carbon dioxide, water that doesn't that represent in the surrounding by using sunlight energy. So under autotropic nutrition, we have two classification: the photoautotropic and chemoautotropic nutrition. Photoautotropic nutrition, uh, the light energy is used to convert simple substances to complex organic compounds. So here in photoautotropic nutrition, they uses water, carbon dioxide, and sunlight to create glucose. Glucose is the organic compound, which is the form of their nutrition. On the other hand, we have the chemoautotropic nutrition, that is the oxidation of inorganic compound that is used as energy source for the conversion of simple inorganic compound to complex organic compound. If there is no presence of sunlight, other organisms tend to use chemicals or other types of element or inorganic compound that is present or abundant in their surroundings, and then they will convert it into organic compounds, so they can create nutrition from sulfur rich environment and methane and many more. Okay, so those are the chemoautotropic nutrition. Here are the examples of photoautotropic and chemoautotropic. So under photoautotropic are plants, so trees, grasses that can be found in terrestrial land. We have the algae, diatom, seaweeds or aquatic area, and bacteria that uses sunlight are cyanobacteria and purple bacteria. Those can be found in aquatic and terrestrial. Under chemoautotropic, they are mostly found where light does not reach. That's why they uses different or inorganic substances such as sulfur, nitrogen, ammonia, nitrates, iron, and phosphorus as the source of their energy to fix carbon dioxide. So this diagram shows how the chemoautotropic organism convert inorganic substances to form sugar as their own nutrition. So this is the process for our photoautotropic nutrition. So they uses carbon dioxide and water, and then with the use of chlorophyll, the sunlight or photons will be captured to form a carbohydrates and produce a byproduct which is oxygen. So here in the process of photoautotropic, light and dark reaction occur. So that's the next is the heterotropic nutrition. It is known to be the mode of nutrition in which certain organisms are dependent on other organisms in order to survive. So heterotropic. Organisms that cannot prepare their own food and have to depend on other organisms known as heterotropic or heterotrophs. So the examples of heterotropic are animals, fungi, and bacteria. Heterotropic nutrition can be classified into three: saprotropic nutrition, parasitic, and holozoic nutrition. So what is the differences between the three? Saprotropic nutrition, they feed on dead or decaying matter. It includes bacteria and fungi, which they just digest the food externally before the nutrients are absorbed. Examples are fungi and bread molds. So they prefer decaying matter or dead matter. Next is parasitic nutrition. Parasitic nutrition is a heterotrophic that obtains nutrients from living organisms. Most of them are labeled as a parasite, so they obtain nutrient by living inside or outside the body of their host. So, example known is cuscuta and tapeworms. Those are they portray a parasitic nutrition. Lastly is the holozoic nutrition. Holozoic nutrition, this is a type of heterotrophic nutrition wherein organisms feed by ingesting a solid organic matter, which is then digest and absorbed into their bodies. Okay, so example is human being and some animals. Here are the heterotrophic nutrition. We have the parasites. Ayan, so those are the endo and exoparasite. As you can see, we have here some tapeworms and then some lice bugs thick. Those are the ectoparasite. The other side is the saprotropic. Saprotropic, they refer decaying matter, no? So most of them are bacteria, fungi, and some are molds. We have different classification of holozoic nutrition. They could be herbivores, carnivores, or omnivores. Herbivores or organisms that feed only on plants. Carnivores, those are organisms that feed on other organism, while omnivores have a choice, so they can have both plants and other animals. In Holozoic nutrition, we're going to discuss the process of digestion of food. So we're going to tackle how nutrition are being absorbed, digest, and excrete uh, in the process of digestion. The digestion in human consists of two types of digestion: the chemical and the mechanical digestion. Chemical digestion uses enzymes to break down food. For example, is amylase. Amylase is an enzyme uh, present in mouth and our small intestine that break down sugar. Next is the mechanical digestion. So this is a process uh, wherein the food are being physically break down of larger pieces into smaller pieces. Example are chewing and churning, chewing in our mouth and then churning in our stomach. So nutrients are absorbed through the wall of our small intestine. So the process of absorption of nutrients starts and happens in small intestine, because here in small intestine the capillaries are present, wherein the diffusion of high concentrated nutrient and low concentrated nutrient are being used to absorb the essential minerals and nutrients to support and to transport within our or different body parts. As you can see, this is an image that shows the villi of small intestine that can be found on the epithelial tissue. So they have a hair-like structure which is known as villi or villus. They consist of a capillaries, okay, or the smallest blood vessels. From that, they will use the red blood cells to carry nutrients throughout our body. So the cellular respiration, obtain energy from sugar, including the sugar from the photosynthesis of plants, organisms process sugar through cellular respiration. The three steps or pathway that convert sugar to an energy molecule is known as the adenosine triphosphate, which cell uses for all their processes. Once we absorb glucose or the sugar in our blood, it will be transported into our mitochondria for us to convert another type of energy, chemical energy, known as ATP or adenosine triphosphate. So that is known as the energy currency of cells. The three metabolic pathway for us to create large amount of ATP are glycolysis, citric acid cycle, or also known as Krebs cycle, and then the oxidative phosphorylation. Okay, so that's the end part of our digestive system. So if you will recall, digestive system starts with our mouth and then followed by the it will be chew, and it will become bolus. Okay, it will be pushed down into our esophagus and then proceed to the stomach. So stomach consists of several gastric juices that helps to digest food furthermore. And then, as you can see, the food will travel into the GI tract or the intestinal tract, followed by the small intestine where the nutrients will be absorbed, and then large intestine. There are some accessory organ such as liver, pancreas, bile duct, bile acid. So those are the accessory organ that helps for the digestion of food. Okay. The last one is the interrelationship among autotrophs and heterotrophs in our ecosystem. So from our discussion, the reproduction of plants and animal nutrition and how they carry out to produce chemical energy, all living organisms are connected in many ways. It could be bacteria, plant cells, animals, human. They are connected in many ways in our ecosystem. One example of this connection is the exchange of carbon between autotrophs and heterotrophs. So as you can see, uh, autotrophs, they uses the photosynthesis to create their own food and releases a byproduct, which is oxygen. O- Oxygen, on the other hand, is essential for animals, which is heterotrophs, for them to break down the food that they eaten. So oxygen is essential for them to proceed in a cellular respiration. So at the end of the cellular respiration, the byproduct is one of the byproduct is carbon dioxide. And that carbon dioxide will be used by the autotrophs. So those type of connection is can be observed in ecosystem, and it is how the autotrophs and heterotrophs connected in many ways. So when we recall, diba, the carbon dioxide is the basic building block that most autotrophs use to build glucose. So the photoautotropic organism harness the energy from the sun and uses it to form the bond that link carbon atoms together while producing oxygen as a byproduct. So that shows the interconnectedness of animals, plants, bacteria. The heterotrophs, especially plant eating heterotrophs, obtain the high energy sugar, which is the glucose, from the breaking them down by respiration, which is the cellular respiration, to obtain cellular energy. And aerobic respiration requires oxygen. Okay, so oxygen that came from autotrophs, from the process of their photosynthesis. Thus, we can see that there is a constant exchange of oxygen and carbon dioxide between the autotrophs and heterotrophs. So that's the end for our week number 1.